Good evening, everyone. Am I audible?
Good evening, ma'am. Yes, ma'am.
Okay. So today we will cover three topics. That is your isolation of the DNA from bacterial cell and isolation of the DNA from human hair and human saliva. Okay. And we have already discussed the topic like your isolation of the DNA from animal tissue, human blood, and your plant leaf, right? These
Yes,
three
ma'am.
sources are completed right now. Isolation of DNA from that animal tissue, human blood, and your plant. And also we covered that topic that is your agarose gel electrophoresis. So till now, anybody have any doubt? Any topic? Okay. Now I'm going to share your screen with you. Then after that, I will explain. Okay. So I will explain like first of all, I discuss one topic. Then show the experimental video related to this. Then after that, I will explain the another topic, right? Now, discuss the first topic that is your isolation of the DNA from the bacterial cell. Just a minute. Just 1 minute. So today, the first topic that we are going to discuss is the isolation of the DNA from bacterial cell. Today, the source is what? Today, the source is this bacterial cell. So, first of all, we have to know that what is this bacterial cell are. These bacterial cell are the colony of the bacteria which we get colony of the bacteria which we get when we pour the media into the sample. Okay, after 48 hours. And it is calculated as CFU, which means that colony forming unit. Now, how we prepare this bacterial cell? So, this will be prepared by two methods. One with the help of the nutrient agar, and another one will be nutrient broth. The colony that we get from the nutrient agar is known as a bacterial cell, and the colony that we get from the nutrient broth is known as bacterial broth. Now, this nutrient agar uh, this nutrient agar are composed of the different type of the chemical like beef and yeast extract, peptone, sodium chloride, and agar. This beef extract or a yeast extract is required around 3 gram per liter, which means that if we have a 1 liter of the solution, then in that case we require 3 gram of the beef extract, not and or. because you can use either beef extract or either yeast extract. Then we require peptone around 5 gram per liter. Sodium chloride is required for 5 gram per liter and agar is required for 15 gram per liter. This agar is act as a solidifying agent. This agar is act as a solidifying agent. And sodium chloride is used to maintain the osmotic balance and it is used to provide the nutrition. Okay, so this is not a part of your curriculum, but I just tell you. Now there is a difference between the nutrient agar and nutrient broth is only that the presence of this agar. Just a minute. So nutrient broth consists of the beef extract, peptone and sodium chloride. But the agar is not present. So nutrient broth is composed of the beef extract or yeast extract, peptone and sodium chloride with same amount. Okay. Now you can do it by any of these methods. Then you have to grow the colony. And the colony that you get for the isolation of the DNA, it should be for at least 24 hours old. That is known as a sample. So how you prepare the sample for the isolation of the DNA from the bacterial cell is clear?
Yes, ma'am.
Okay. Now the next is your reagent required for this process. The first reagent is your lysozyme. It is a enzyme which is used to break the cell of the bacteria. It is used to break the cell wall of the bacteria. Then after that, we required phenol chloroform solution. This solution is required around 1:1, which means that if we required 1 ml of the phenol, then we required 1 ml of that chloroform. It is used to remove the protein and carbohydrate. It is used to remove the protein and carbohydrate. Sometime in the place of this phenol and chloroform, we can also use the potassium acetate for this purpose. Potassium acetate in the place of this phenol and chloroform solution. We can also use what? We can also use potassium acetate so that this protein and carbohydrate can be removed. Okay. Then next is your SDS It acts as a detergent source. Next is your sodium acetate. Sodium acetate is used to remove the histone protein. Next is your chilled ethanol. Of 70%. It is used to separate the DNA molecule from solvent. Then we use TE buffer. TE buffer is what? It is tris EDTA buffer. It is used for the storage. Screen is visible.
Yes, ma'am.
Just a minute. Just give me 1 minute. It is visible now.
Yes, ma'am.
Okay. Now this TE buffer is used for the storage of the DNA molecule. And we required a broth culture. And this broth culture is what? This broth culture is for the, at least it is 24-hour broth. Now this TE buffer is prepared by the previous one that we already prepared it. Okay. SDS is prepared by 12%. Like if you have to prepare it in 100 ml, then what you have to do, 12 by 100 multiplied by 100, that is 12 gram in 100 ml. Sodium acetate is prepared for 1 molar. So you can use the formula, that is weight is equal to molecular molarity multiplied by molecular weight multiplied by volume given divided by 1000. Then this chilled ethanol 70% is again, it is prepared by 70 by 100 multiplied by 100, that is 70 ml into 30 ml of the distilled water, and the ratio of phenol and chloroform that I already told you. So how we prepare this reagent is clear. Preparation
Yes,
of
ma'am.
the reagent, sorry, preparation of the reagent is completed. Next is your role of the chemical is also completed. And after that, we are going to see that the procedure. So for procedure, we required 5 ml 24-hour broth culture. Okay. Now, if you have a broth cell, sorry, culture cell, now you have what you have to do. You have to dissolve that sufficient amount of the cell into the 5 ml of the distilled water. Then centrifuge it 3000 RPM for 10 minutes. Okay, then you get what? You get supernet and palette. You have to discard that supernet, and in that palette you have to add 2 ml of PE buffer, that is your tris EDTA buffer, plus 70 microliter, 75 microliter 75 microliter 12% SDS plus 7.5 microliter lysozyme. Then you have to incubate it at 37 degrees Celsius for 1 hour. Then after that, you have to add 3 ml of chloroform and phenol solution. Then you have to centrifuge it. Sorry, not centrifuge, you have to refrigerate. After adding of 3 ml of that chloroform and phenol solution, you have to refrigerate it for 1 hour. Refrigerate it for 1 hour. Then you have to do a centrifuge at 5000 RPM for 15 minutes. Then you get two type of the layer. One is your normal layer and another one is your aqueous layer. So you have to transfer that aqueous layer into the another append drop, and you have to add 3 ml of chloroform plus phenol solution. Then you have to again refrigerate it for 15 minutes. Then after refrigeration, again you have to centrifuge it. This time you have to centrifuge it at 5000 RPM for 15 minutes. Then again you get a upper aqueous layer and the another one is your normal layer. Again you have to transfer it into the another tube and add 1/10 volume of sodium acetate plus just double chilled 70% ethanol, and you have to resuspended it with the TE buffer around 50 to 60 microliter and refrigerate it. Fibrous cotton-like structure is known as a DNA molecule. Fibrous, cotton-like structure known as a DNA molecule. Clear my points? Isolation of the DNA from bacterial cell is clear.
Yes, ma'am.
Okay. Now, isolation of the DNA from human hair will also be explained, and after that, I will show you the video.
मैम
Yes,
मैम हम लोग यूज क्यों करते हैं क्लोरोफॉर्म का?
क्लोरोफॉर्म और फिनॉल सोल्यूशन का।
यस मैम।
ये देखिए, मैंने आपको यहाँ पे बताया था ना कि फिनॉल और क्लोरोफॉर्म का सोल्यूशन जब हम यूज करते हैं तो जो प्रोटीन और कार्बोहाइड्रेट प्रेजेंट होता है बैक्टीरियल सेल में, तो वो रिमूव हो जाता है। देखो, हमें जब डीएनए को आइसोलेट करना है तो मेन एम हमारा क्या है कि हमको सिर्फ डीएनए मिले, बाकी जितने भी कंपोनेंट है वो सब कुछ रिमूव हो जाए। तो फिनॉल और क्लोरोफॉर्म का सोल्यूशन दोनों मिलकर जो प्रोटीन और कार्बोहाइड्रेट प्रेजेंट होते हैं बैक्टीरियल सेल में, वो उसको रिमूव कर देता है। क्लियर?
ओके मैम।
क्या सबको इंग्लिश में कोई प्रॉब्लम हो रही है? हिंदी में डिस्कस कर लो। प्लीज टेल मी इफ यू वांट टू डिस्कस
मैम
इन हिंदी।
थोड़ा थोड़ा कर सकती है मैम।
ओके, योर नेम?
वरुण सिंह।
वरुण। ओके वरुण। नाउ द नेक्स्ट इज isolation of the DNA from the human hair. Okay, so when we have to isolate the DNA from the human hair, we have to prepare this sample. Sorry, we have to collect this sample. Then the collection of the sample is according to the selection criteria. Isolation of DNA from human hair. Okay, so sample selection is what? Sample selection is very important. The sample should be soft, new tissue, easy to handle, easy to grind, plus easy to cell lysis from the free from the contamination. Easy to cell lysis and free from contamination. Okay, now what we have to do, take a hair, and cut into the small strand and keep it at petri plate. Keep it at petri plate. After that, transfer it into the mortar pestle. Plus add 10 ml of lysis buffer. Add it slowly to make a paste. Otherwise, slurry is formed. Then filter it and transfer the liquid part into apple drop. Lysosine buffer is used to break the cell wall of the, break the cell of the human hair. Lysis buffer is what? It is used to break the cell of the human hair. Now, after transferring it into the apple drop, add 100 microliter of proteinase buffer. Proteinase K, proteinase K, and centrifuge it at 5500 rpm for 10 minutes. Here we add a proteinase K to remove the protein. Then we get what? We get a supinant and palate. Discard that supinant, and in that palette you have to add 600 microliter of chloroform and isoamyl alcohol. Isoamyl alcohol. Okay. Here the ratio of the chloroform and isoamyl alcohol is 20:1, which means that if you take 24 ml of that chloroform, then you have to add 1 ml of the isoamyl alcohol. Then you have to mix it properly. After mixing it, again centrifuge. This time you have to centrifuge it again at 5500 rpm for 10 minutes. Then you get what? You get supinant and palate. Discard that supinant and into the palate you have to add 600 microliter isopropanol and invert it for 5 minutes for proper mixing. For proper mixing. Then centrifuge it at 10,000 rpm for 10 minutes. Then again supinant and palate. Discard that supinant and into the palate you have to add 600 microliter chilled ethanol. Mix it properly. Then centrifuge at 10,000 rpm for 15 minutes. Then you get what? You get supinant and palate. Discard that supinant. Palate you have to air dry so that it remove the smell of ethanol. And after that, you have to add 10 microliter, sorry, 100 microliter of TE buffer for storage purpose. Then refrigerate it. Clear? Isolation of the DNA from human hair is clear? वीडियो दिखाऊं?
Yes, ma'am.
ओके पहले दिखाऊंगी isolation of the DNA from bacterial cell, then after that isolation of the DNA from human hair. If voice is not clear, then let me know, okay?
Hello everyone, welcome to the ITEL Academy. Today we are going to perform isolate
Just 1 minute.
extraction of DNA from bacterial cells. So these are the chemicals which are required for performing extraction of DNA from bacterial cells. The very first is 10% solution of SDS. SDS is what? Sodium dodecyl sulfate. Phenol, chloroform, lysozyme, and buffer solution, distilled water, and with that we also need the solution of sodium acetate. Hello everyone, welcome to the ITEL Academy. Today we are going to perform isolate extraction of DNA from bacterial cells. So these are the chemicals which are required for performing extraction of DNA from bacterial cells. The very first is 10% solution of SDS. SDS is what? Sodium dodecyl sulfate. Phenol, chloroform, lysozyme, and buffer solution, distilled water, and with that we also need the solution of sodium acetate. Okay. So first of all, what we are going to do, we need to, the very first step is take 1 ml of distilled water in the appendrof, and see, you can see we have already marked this in the pipette, and now this is the appendrof. Now I'm going to pour the water, measured water in this. And we need to fill all the 12 appendrof with the same amount of distilled water. Here we have already filled each appendrof with 1 milliliter of distilled water, and as you can see I already mark all the appendrof with B. See, B is indicating that we are extracting the DNA from bacterial cell. Now with the help of inoculating loop, I'm going to extract the bacteria from our culture and pour it into the appendrof for making culture broth. So by the same procedure, we need to fill all the 12 appendrof, and after that we need to centrifuge all these appendrof at 3000 rpm for 10 minutes. Okay. So as you can see here, our appendrof is ready with the bacteria. As you can see it here, is it visible to you? See, you can see here, this is the white color bacteria. So we already inoculate the bacteria in the appendrof. Now by the same procedure, we need to perform all these 12 appendrof. After filling the bacteria into the appendrof, now you can see these white pellets clearly. Okay, what we are going to do, we are going to need to centrifuge at it for 10 minutes at 3000 RPM, as you can see. Now place all the pin drops carefully and make sure that you can balance it properly. Here you can see the mouth of each pin drop is it in the towards the center. So now here we are ready to go. As you can see the time, it's 10 minutes and 3000 revolution per minute. So now I'm going to start it. After 10 minutes you will see the result. So friends, after performing the centrifugation, now we have take it out from the centrifuge. What we are going to do now, we need to discard the supernatant and we take further pellets. Okay, so for discarding, you remember that pellet should not disturbed while you discarding the supernatant. So here you can say discard the supernatant and now only the pellets are remain there. By the same procedure we need to discard all the append drops and after that we work with the pellets. Okay. So after discarding all the supernatants, the pellets are only remain here. Now very first step is we need to pour this tea buffer, 0.5 ml of tea buffer in each of the append drops, and for that I set 500 microliter on this. Now with the help of micro pipette, take 0.5 ml of tea buffer and add it into each append drop, and by the same measurement we need to add this buffer solution in each append drop. So after adding 0.5 ml of tea buffer to each append drop, now what we are going to do, we need to add 75 microliter of a 10% SDS solution in each append drop. So after adding 75 microliter of 10% SDS to each append drop, now we need to add 7.5 microliter of lysozyme to each append drop. So here we have already set 7.5. Now I'm going to take 7.5 per microliter of lysozyme and pour it into each append drop. Okay, by the same procedure, we need to add it all the append drops. So after adding 7.5 microliter of lysozyme in each append drop, we need to incubate at room temperature for at least 1 hour. Okay. So after incubation at 37 degrees Celsius for 1 hour, now we are going to add 0.5 ml of chloroform and phenol in each append drop. So I already set it at 500 microliter. Now I'm going to add 0.5 ml of chloroform in each append drop. So here we go.
Yes, ma'am.
Now in the same append drop, we need to add 0.5 ml of phenol. So by the same amount, the same amount we need to add in each append drop 0.5 ml of chloroform and 0.5 ml of phenol. So one more thing which I would like to add, that you need to boil the phenol in water bath. So after adding chloroform and phenol to each of the append drop, now we need to refrigerate these append drop for 15 minutes, and after refrigeration we further need to go with centrifugation at 5000 rpm for 15 minutes. So after centrifugation we are going to saw the result. So after refrigeration for 15 minutes and after performing the centrifugation, now we need to discard the supernatant of each append drop, and after discarding the supernatant of each append drop, we need to add further 0.5 ml of chloroform and 0.5 ml of phenol in each of the append drop, and further refrigerate it for 15 minutes, and after performing the refrigeration we need to undergo centrifugation at 5000 rpm for 15 minutes. But you need to make sure that while you're dealing with the phenol, you wear proper gloves, mask, and all these things. It would be helpful for your health. So after performing the centrifugation, now I am going to discard the supernatant, and we need to carefully discard the supernatant so that pellet should not damage. So after discarding the supernatant, now I am going to add 100 microliter of sodium acetate in each of the append drop. So during discarding, we need to make sure that pellets should not be damaged. As I already set on micropipette 100 microliter, now I am going to add this amount in each of the append drop. We need to make sure that we can pour this amount very carefully in each of the append drop, and after that I am going to add 200 microliter of chilled ethanol in each of the append drop. So as you can see, I am pouring sodium acetate in each of the append drop. So after adding 100 microliter of sodium acetate in each of the append drop, now I am going to add 200 microliter of ethanol in each of the append drop. And after adding 200 microliter of chill ethanol in each of the append drop, we need to keep these append drop for few minutes, and after that we need to discard the supernatant. So after adding chill ethanol, now I'm going to discard the supernatant, and after discarding the supernatant, I'm going to add 50 microliter of tea buffer in each of the append drop. आपने pause दिया होगा. So after discarding the supernatant, I am going to add 50 microliter of TE buffer, and after adding this TE buffer, we are going to store the append drop. So after adding 50 microliter of tea buffer in each of the append drop, now our process of DNA extraction is completed. Now we are going to store these in the refrigerator for performing the further processes like PCR, gel electrophoresis, etc. Hope you got it.
Yes, madam.
Yes, ma'am.
Isolation of the DNA from living here.
Hello everyone, welcome to the ITLS Academy. Today we are going to perform isolation of DNA from human hair sample. So here we are having the human hair sample. Now what we are going to do, we need to make the paste of this hair sample, and for this we need to add 10 ml of lysis buffer in this, and with the help of mortar and pestle I'm going to grind this hair sample and need to add drop by drop lysis buffer so that we can make the paste, because if we add the entire amount in a one time it become a slurry, and which we cannot further use for the experiment. That's why we need to make a paste to grind it properly. After grinding it properly, now we need to filter it out. So here, by using filter paper, I'm going to filter the slurry. Here you can see. So after filtering the slurry, I am going to pour 500 microliter of this slurry into each of the apple drop. As you can see. I already set the number on the micropipette, that is 500 microliter. Now we are going to add protease K into each of these apple drop. Do you know why we add proteinase K? Just so that the protein which is present in it can be degraded. Have we stopped? So after adding 500 microliter of slurry into each of the apple drop, I am going to add 100 microliter of protease K in each of the apple drop with the help of micropipette. Protease K is added just because of for the digestion of protein which is present in the sample. And after adding this protease K in each of the apple drop, we need to centrifuge it at 5500 rpm for 10 minutes. So after centrifugation, as you can see, there is a pallet, and this is our supernatant. So what we are going to do, we need to discard the supernatant very carefully. Again, we need to discard supernatant very carefully so that pallet should not be destroyed. So now after discarding the supernatant, I am going to add a solution of chloroform and isoamyl alcohol. For this is a ratio, the ratio of chloroform and isoamyl alcohol is 24:1. For preparing this solution, I'm add 12 ml of chloroform and 0.5 ml of isoamyl alcohol. So now after preparing this solution, we need to add 600 microliter in each of the apple drop. Now after adding this solution of chloroform and isoamyl alcohol, we need to invert the apple drop for at least 10 minutes, and after that we need to centrifuge at 5500 rpm for 10 minutes. So after performing the centrifugation for 10 minutes, now what we need to do, again we need to discard the supernatant and add 600 microliter of isopropanol in each of the apple drop. Yeah. So as you can see, these are the pellets and this is the supernatant. Now again I'm going to discard the supernatant. Discard it very carefully. Pellet should not be and after discarding the supernatant, we need to add 600 microliter of isopropanol. So here I'm going to add 600 microliter of isopropanol in each of the append drop. Now after adding isopropanol in each of the append drop, we need to invert it at least for 5 minutes, and after that we need to perform centrifugation at 10,000 rpm for 10 minutes. So after performing the centrifugation, again we need to discard the supernatant and palate should not be harmed. And after discarding the supernatant, we need to add 70% of chill ethanol, 600 microliter of 70% chill ethanol in each of the append drop. Start now. Go. So here I have already set on micropipette 600 microliter. Now I'm going to add 600 microliter of chill ethanol in each of the append drop. Now after adding this 600 microliter of chill ethanol, invert it for 5 minutes and again centrifuge at 10,000 rpm for further 10 minutes. So after performing the centrifugation, as you can see there are pallets and the supernatant. Now what I'm going to do, again the same procedure, we need to discard the supernatant. But one additional thing which we need to do here is that we add 70% of chill ethanol before centrifugation. Now there is a smell of ethanol in the pallet. So what we need to do, we need to evaporate the smell. Until then we need to keep them open, and after the smell it will be evaporated, we need to add fresh EDTA buffer in this. So after 30 minutes the smell is evaporated. Now I'm going to add 100 microliter of fresh EDTA buffer in each of the append drop, and we can stop the process after adding fresh EDTA buffer. Hope you understand the entire process. If you are having any doubt, just let me know in the comment box. Thank you so very much.
After that, we discuss one more topic, that is here, isolation of the DNA from human saliva. So this is very easy. We don't require any chemical for this, okay? We just simply rinse it, and then after that we centrifuge it in a repeated manner when we get the DNA. So first of all, I will explain you, then I will show you the experimental video. Isolation of DNA from human saliva. For this, what we have to do, we take a water and add salt into it. Okay. Then we take it into the mouth. Take it into the mouth. Then we rinse it for 10 minutes. After that, we transfer it into the juicer and centrifuge it at 12,000 rpm for 2 minutes. And we get what? We get the supernet and salad. We discard that supernet and we repeat this process. Like again, we transfer it into the juicer and take into the append drop. Then after that we centrifuge it. Okay, so we repeat this step until we get a clear white color palette. White color palette. Okay, this is the But again, we transfer it into the upper drawer and centrifuge it at 12,000 RPM for 2 minutes, and again the discipline. And in pilot, again we added that water which are present into the beaker. Again we do that, and we repeat it until we get a clear white color palette. Okay, when we get the clear white color palette, then we added a 100 microliters of CE buffer for the storage and store it at a refrigerated condition. Clear my point?
Yes, ma'am.
And I will show you the video related to this. This is very easy.
Hello everyone, welcome to the ITLS Academy. Okay, before starting anything, do you know what is DNA? Okay, let me tell you. DNA is a deoxyribonucleic acid, which is the genetic material of ours, basically human genetic material, and it is hereditary, which can be transferred from our parents to our offspring. So we can say from generation to generation, due to which we can see variations in us. Okay. So today we are here to perform the isolation or extraction of DNA from human saliva. Okay, for that we required, these are the material which we required for performing, like micropipette, reagent. So this is the material which we require for performing isolation of DNA from human saliva. This is Epindrof, and this is our Tris EDTA buffer, which acts as a buffer solution. This is micropipette, and these are test tube holders, and water, obviously, for which we need to rinse. So first of all, for the very first step is we mark on the Epindrop. You can see here I mark the first word of my name. This is T. So first of all we need to mark the Epindrop. Secondly, what we need to do, we need to take a sip of water and we need to rinse it up to 10 minutes. So this is the rinse water after 10 minutes. Now what we need to do, we need to transfer 1.5 microliter of this solution into every Epindrop. We set the micropipette on 500 and now we are going to take the solution and transfer it into each Epindrop. By the same procedure, we need to transfer the solution into each Epindrop. Now we already transferred the solution into each Epindrop. First, now we are going to perform centrifugation, and we need to perform centrifuge for the 2 minutes at full speed. Now you need to keep all these into the centrifuge. You need to make sure that you can put these Epindrop in the balance.